At Plasmidsaurus, we offer flexible microbiome sequencing services for any microbiome workflow. We have a couple of options for how you can prepare your sample. You can send us raw biological samples and let us do all the processing. Check out that video to see those instructions. You can send us 16S, 18S, or ITS amplicons that you've PCR'd yourself. Or you can send us genomic DNA preps and we'll amplify the 16S genes. In this video, we'll walk you through the process of sending us amplicons or gDNA for sequencing. If you send us genomic DNA, we'll sequence the 16S region for microbiome profiling. We recommend using a commercial column-based genomic DNA extraction kit. Remember to include an RNase treatment step in your extraction to remove any RNA. Once your extractions are complete, you'll want to check that you have enough genomic DNA with a fluorometric method like a qubit. You'll get the most out of long-read sequencing by sending highly pure, high molecular weight gDNA. You can check these with spectroscopy and electrophoresis. Once you've quantified and characterized your DNA, normalize it to the concentration indicated for the service level you chose. Another option for microbiome profiling is to prepare the 16S, 18S, or ITS amplicons yourself. Amplify 16S, 18S, and/or ITS DNA according to your preferred protocol. You can purify your amplicons yourself, or let Plasmidsaurus handle the PCR cleanup for you. Just click one box and we'll take care of the rest. Check your sample's DNA concentration with a fluorometric method like a qubit and normalize to between 10 and 20 nanograms per microliter. On the Plasmidsaurus website, choose an order name and a name for each of your samples. You'll get a 6-character order ID that you'll use to label your tubes. For orders of 48 samples or less, use 8-strip tubes. The first 2 characters of your order ID go on the first tube of each strip. The other tubes are numbered in order. For more than 48 samples, you can use 96 well plates with 200 microliter wells. Label the front of the plate with the full 6-character order ID. Label either the first row or the first column with the sample numbers. If you're sealing your plates manually, make sure you use a plate roller and really push down hard to prevent leakage during shipping. Or, even better is to use a heat sealing device if you have one. For 8-strip tubes, use a falcon tube or some bubble wrap to protect your sample during shipping. Ship your wrapped samples inside a plastic bag. For samples in plates, wrap each plate individually and avoid stacking plates because the plates on top can damage the seals of the plates on the bottom. Print out a copy of your order form and put it inside the bag so that the QR code is visible. And you're ready to go. Drop off your samples in your nearest Plasmidsaurus Dropbox. The microbiome sequencing results will be ready as soon as the following morning. I get an automatically generated microbial community profile for each sample. It shows the most common species and their relative abundance. If I want to go deeper, I can download the full report with all the raw reads and taxonomy labels. And that's how we do microbiome sequencing. Thank you, Plasmidsaurus.